Hello there and welcome back. This is our third video in our video series for Chapter 8 on recombinant DNA technology. Once again, I'm Miss Delia and let's get started with the lesson. In the previous videos, we talked about what are the tools in recombinant DNA technology and the methods to apply them in gene cloning. And in this video, we're going to go into 8.3, the application of recombinant DNA technology. So basically, how to use what we have learned so far. In addition to that, we also have the learning outcomes for this subtopic. So I hope that by the end of this lesson, you should be able to briefly explain the applications of recombinant DNA technology in mass production of insulin using cDNA, and also describe the steps in the production of insulin using cDNA. So it looks like we have a focus in this chapter and we need to learn something new, which is cDNA. We'll go into that in a little bit. But first, let's talk about the many ways that we can use recombinant DNA technology. So after we learned about how genes are, how DNA works, and how to turn from DNA to mRNA, mRNA to protein, we learned to apply it in science and to make it useful for us, basically. So there are three main categories that I'm going to talk about how we apply recombinant DNA technology. Macam mana kita gunakan ilmu yang kita belajar pasal DNA ini untuk memanfaatkan kita. Ada tiga kategori utama yang saya akan fokuskan. Okay, so the first category, we're going to talk about agriculture. Bila kita cakap pasal pertanian dan penternakan, okay, kita ada dua contoh utama. Okay, there's two main examples that we usually use. The first example is you can use recombinant DNA technology to create transgenic plants. So, kamu boleh cari gene yang boleh memanfaatkan tumbuhan yang kamu mau tanam. Okay, so you take that gene, you put it into the plant that you want to grow, and then basically you are going to get plants that can be more resistant to herbicides. So, kalau kamu tanam ini di padang, okay, atau di kebun kamu, ada rumput-rumput rumpai lain. Okay, so say example, you want to spray them with herbicides, kamu mau sembur dengan racun, kamu pilih gene untuk menyebabkan tanaman kamu rintang racun, tapi rumput lain kamu tidak bagi gene itu. So, bila kamu sembur racun, rumput lain akan mati, tanaman kamu masih hidup. So, that is an example of how we can use this in plants such as soybeans. Contoh kacang soya, kita boleh bagi gene ini supaya dia lebih rintang racun. We can also use the same method to make plants more resistant to pests. Okay? So, kita boleh juga membolehkan tumbuhan-tumbuhan yang kita tanam itu supaya lebih rintang serangga perosak. So there are many ways. We can learn about that in another time. And another example in agriculture, tadi itu pertanian, sekarang kita cakap pasal penternakan, is creating transgenic animals. So you can get genes that will make certain that will make your organisms grow faster. Okay, so for example, we take this gene, this growth gene, and then we put it into salmon, the fish. Okay, so say for example, you take that gene that makes things grow faster and then you put it into a fish. That means this fish will now take the same amount of time but grow even bigger. Okay, so an example of animal that we use this method on is salmon. Contoh ikan salmon yang selalu warna merah atas sushi itu, okay, kita boleh gunakan method ini. So that, for example, in 1 year, we can get salmon that is this big. Normal salmon without recombinant DNA technology, after 1 year, maybe this big. But ours, with the transgenic salmon, after 1 year, it grows much bigger. That means we have more food in a shorter amount of time, and that can be better for us because more profits, more food, everybody is happy. Okay, so those are two examples in agriculture. Another example of application of recombinant DNA technology but in a different category is in the production of insulin. So kita boleh gunakan apa yang ilmu kita belajar pasal recombinant DNA technology dalam bidang kajian dan perubatan. Contoh dalam bidang kajian dan perubatan yang kita akan gunakan di sini adalah penghasilan hormon insulin. So insulin is the hormone that is necessary to regulate the amount of sugar that is in your blood. Orang yang diabetic, people who are diabetic, they have a very, very low level of insulin. Since what I produce in my body is just enough for me, you cannot take insulin from me and give it to somebody who is diabetic. Okay? Kita tidak boleh kasih keluar daripada manusia. Tidak cukup untuk orang yang berpenyakit diabetik tu. So what we can do is you can take insulin from me, take it to the lab, produce it in the lab, and then sell this to people who are diabetic. Okay, kita boleh ambil gene daripada badan kita, hasilkan insulin dalam lab, dan gunakan insulin yang dihasilkan itu berikan kepada orang yang berpenyakit darah tinggi, kencing manis. Diabetic is kencing manis. So yeah, so that's one of the applications. And we're going to go more into production of insulin later because we are going to learn how we do that by using cDNA. Before we go into that stuff, let me finish about other applications of recombinant DNA technology. So kamu nampak di bawah, number 3, environment. Examples of how we can use recombinant DNA technology for the environment is we can use it to create bacteria that can clean up or degrade oil spills. So ayat di bawah tu, degradation of oil spill adalah menguraikan minyak-minyak yang tertumpah di laut. So you can learn more about oil spills later. I'm telling you that the technology for this is becoming more developed now. I have a cousin who is in UMP right now, University Malaysia Pahang, and they are researching on how bacteria can digest certain types of plastics. And other universities around the world, other research centers are finding out ways on how we can create transgenic bacteria to clean up oil spills, to digest plastic, to clean up certain wastes that are in our environment. Basically, to clean up the effects of pollution that we did. Another method that, or another application of how we use recombinant DNA technology to alter our environment is to create transgenic mosquitoes. So if you remember a few years ago, there was the uproar of the Zika virus. So one of the methods that they are trying to overcome the Zika virus is by producing transgenic mosquitoes. We have a lot of diseases that are transmitted via mosquitoes. So if we produce transgenic mosquitoes that are sterile, will not produce baby mosquitoes, then we can essentially eradicate these diseases. Ada penyakit yang datang, merebak disebabkan nyamuk, kita hasilkan transgenic mosquito, nyamuk yang sterile, yang mandul. Okay, so the nyamuk cannot make baby nyamuk, dan tiada jentik-jentik, tiada nyamuk baru, penyakit itu pun tidak akan merebak. So those are the examples of applications of recombinant DNA technology. So this diagram is also a refresher and telling you more about certain examples. So di sini adalah untuk ingatkan balik macam mana cara kita buat gene cloning menggunakan plasmid sebagai vector, and these are some of the applications. So in here you could see that I talk about using the genes for pest resistance, put that in plants. For agriculture, you can use it to create bacteria that can clean up toxic waste, second picture there. And another application is we can use it to produce proteins in the lab. Those proteins can be used to dissolve blood clots, to treat people who have heart attacks because of blood clots, and we can also produce human growth hormones to help those who are stunted in their growth. So if you have any problems regarding this, feel free to search for information online, ask a friend, comment below, or ask me directly. Okay? Kalau ada yang kamu bingung, pat ni kamu tidak perlu hafal sangat, silalah belajar lebih, dia akan membantu kamu kalau kamu pilih untuk terus mencebur bidang biology di university. Okay, so that is a lot of talking from me. Okay, itu sahaja untuk overview application of recombinant DNA technology. So, benda yang saya cakap dari tadi adalah pelbagai cara untuk menggunakan ilmu yang kita dapat pasal recombinant DNA technology. Now, I am going to focus on one of the applications. Sekarang saya akan tekankan kepada production of insulin using recombinant DNA technology and focusing on cDNA. So if you're ready, prepare yourself. We are going to learn. Okay. Application in production of insulin. First things first, you need to learn what is cDNA. Kita belajar dulu apa itu cDNA. cDNA stands for complementary DNA. What's that? Complementary DNA is a double-stranded DNA made in vitro using mRNA as a template and a reverse transcriptase enzyme. Mari kita tengok. cDNA adalah DNA yang double-stranded, dihasilkan in vitro, yaitu dalam lab, using mRNA as template, menggunakan mRNA sebagai template, and uses the enzyme reverse transcriptase. Menggunakan enzyme reverse transcriptase untuk salin dan hasilkan dia kepada DNA. cDNA lack introns and they can be used directly to express the protein encoded by them. Dalam cDNA ada exon sahaja, tiada intron, dan boleh terus digunakan untuk menghasilkan protein yang diperlukan. This cDNA is often used to clone eukaryotic genes in prokaryotes. So example, we want to take human genes and put it into E. coli, then we need to use the cDNA version. cDNA is used because it represents the expression of the genes without the introns. Remember, eukaryotes have introns and exons in their genes. But bacteria do not have introns. Okay, bacteria punya DNA tiada intron. Dan dalam bacteria tiada RNA splicing. Since there is no RNA splicing in bacteria cell, if you give the bacteria a piece of gene that has intron and exon, it doesn't know where to cut. Okay, kalau kamu bagi gene yang biasa, bacteria tidak tahu mau potong di mana. Jadi, apa cara dia? Kita bagi bacteria DNA yang sedia untuk diguna. We give the bacterial host a ready-to-use DNA, which is cDNA. Kita potong sedia bahagian intron itu, bagi dia exon sahaja. Bila bakteria itu dapat, dia boleh terus translate. Okay, so let us talk about the steps in using cDNA. First things first, we are going to make the cDNA. We are going to take mRNA for insulin from the human, okay? mRNA from insulin is isolated or extracted from the cells of the human pancreas. Kita keluarkan daripada manusia dan kita masukkan ke dalam test tube, okay? And then you are going to add restriction enzymes. Dalam ini adalah mRNA untuk insulin. So what we're gonna do is we're gonna take the restriction enzymes. We're going to add that to cut the mRNA that is in my test tube. Okay, after we add the restriction enzyme to cut the mRNA, we are going to add reverse transcriptase enzyme. So kita ambil reverse transcriptase enzyme and we're going to add that in the test tubes. Gochang. Okay, so reverse transcriptase enzyme is added to synthesize a cDNA by using mRNA as a template. Di sini sekarang akan ada molekul yang separuh mRNA, separuh cDNA, as you can see in this part of the diagram. Oh, hold on, let me put my hand. As you can see in this part of the diagram. So right now in my test tube, I have this thing. Yang separuh pink, separuh biru. After that, I am going to get rid of the mRNA. To get rid of the mRNA, I am going to use mRNA degrading enzyme. Enzyme yang menguraikan mRNA. So I take that mRNA degrading enzyme, saya tuang ke dalam test tube. Goncang, goncang, goncang. So after I add the mRNA degrading enzyme, the pink mRNA strand will be gone. And what is left is single strand cDNA. After this, to make sure that the single strand cDNA will become double strand DNA, I am going to add DNA polymerase enzyme. So sekarang saya tambah DNA polymerase enzymes. Okay. Apa yang terjadi dalam test tube ini adalah dia akan menghasilkan second strand of cDNA. It's going to synthesize a second DNA strand that is complementary to cDNA in vitro. Okay? So daripada tadi saya tambah, daripada mRNA dia akan menjadi separuh mRNA, separuh cDNA. Kita uraikan mRNA dan hasilkan second strand of cDNA. Okay, so I hope that is clear because this is basically how we produce cDNA. Ini adalah cara kita hasilkan cDNA. Alright, so now let's try to use it. Okay, now we try to use the cDNA. We're going to use this cDNA of gene that has no introns. Isolate it from its source and then cut it with the restriction enzyme. In this example, we are going to use restriction enzyme BAMH1. Okay, kita gunakan enzyme BAMH1. Kita tambah ke dalam container yang mengandungi cDNA tadi. And then we are going to remove plasmid from a bacterial cell. Okay, we take the plasmid and then it is opened up at a specific point using the same restriction enzyme. So in here, I will have my plasmid. Ada plasmid di dalam. Saya potong dengan restriction enzyme yang sama. Okay, so mix, mix, mix. The enzyme is going to cut the plasmid inside. DNA is now attached to the plasmid using DNA ligase. Sekarang saya kasih campur. Okay, kasih campur. Now the cDNA is mixing with the plasmid. And then I will add DNA ligase. Campur DNA ligase. Campur, 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 campur. Now in here I have plasmid that have the cDNA gene, which is now called recombinant plasmid. So, disebabkan saya sudah campur cDNA dengan plasmid di dalam ini, sekarang ini mengandungi recombinant plasmid that carries the human DNA for insulin. Now, I can use this in the next step. Now, this recombinant DNA is inserted into the host, which is E. coli. Take this. In here, contain E. coli. Ada E. coli kecil-kecil di dalam ni. Saya campurkan recombinant plasmid ke dalam. Okay. Under the right conditions, the E. coli in my solution will take up the recombinant DNA by transformation. Okay. Ada E. coli yang akan ambil recombinant plasmid tersebut. Dan kalau dia berjaya, itu adalah proses transformation. Now, the bacterium E. coli with its recombinant plasmids is allowed to reproduce. Saya benarkan dia membiak. Finally, a lot of these bacterial clones carrying many copies of the gene of insulin will be produced. Selepas saya biarkan membiak, akan ada banyak E. coli yang mengandungi gene insulin untuk yang digunakan untuk menghasilkan protein nanti. Okay, so yeah. So I'm gonna let these bacteria grow, and when it is time, I will extract the insulin protein from here, and I can use that insulin to make medicine, and that medicine will be used for people with diabetes. Okay? So once again, this is a diagram to kind of summarize and illustrate the steps in using cDNA. All right. Okay, so mula dari sebelah kanan ni, cDNA dihasilkan dengan cara ni, dimasukkan ke dalam plasmid untuk jadi recombinant plasmid, recombinant plasmid dimasukkan ke dalam E. coli, E. coli membiak dan menghasilkan insulin protein. Okay, so that is pretty much it for production of insulin using cDNA. There are some ethical issues when it comes to this topic. Walaupun ilmu ini berguna dan boleh memanfaatkan kita, ada isu dari segi etik dari pelbagai sudut lah. Okay, so the first issue when we talk about recombinant DNA technology, not just for insulin, but also in agriculture and a bunch of other stuff, is GMO. Mesti kamu pernah dengar term ini juga, genetically modified organism. Disebabkan kita hasilkan organisme-organisme ini dalam lab, kita yang pandai-pandai buat, ramai orang yang takut. Mereka takut organisme yang diubah sesuai menggunakan teknologi ini, genetically modified organism, mereka risau organisme ini tidak selamat untuk dimakan. That is an issue some people talk about. Another issue is that recombinant DNA technology is not good in terms of your morals or religious views. So dari pandangan agama, bila kita edit organisme macam ini, macam kita main-main, macam kita buat-buat diri kita ini Tuhan. So that is meant by playing God. Playing God by changing the genetic constitution of an organism. So dari sudut agama, dia macam we are doing something we are not supposed to do. Macam kita pandai-pandai melangkaui batas kita sebagai manusia. And another issue is that in terms of genetically modified things, it can only benefit the rich, okay? Dari segi masyarakat, dari pandangan masyarakat, ada kemungkinan kebaikan-kebaikan ini hanya diberikan atau dirasai oleh negara-negara yang lebih kaya. So, I don't know, for example, in North America or in Europe, they would be able to enjoy these things, but here we are not enjoying it. Or, within a society, there will be rich people, there will be poorer people, there will be middle class people. These recombinant DNA technologies may only be beneficial for those who are rich. Mungkin dia boleh memanfaatkan mereka yang kaya sahaja dan tidak dapat memanfaatkan mereka yang miskin. So itu adalah antara isu-isu ethics yang dibahaskan, dibincangkan, digaduhkan dalam bidang recombinant DNA technologies. Okay? And this doesn't just apply to production of insulin, it applies to many things as well. I think what we can add is that sometimes it's not about who is rich and who is poor, but also what the company decides to do. Okay, kadang-kadang ini boleh disalahkan kepada company-company yang buat research ini juga lah. Kadang-kadang mereka yang pilih siapa yang mereka mau bagi kebaikan ni, siapa yang dia tidak mau bagi. Okay, so that's another thing. And another ethical issue is the environmental impact. So, kalau kita ternak tumbuhan-tumbuhan transgenik itu di kebun sendiri tidak apa, tapi kadang-kadang benih dia boleh terpasing kepada kebun orang lain, terpasing termasuk hutan. So, we think about the unintended transfer of transgenes through cross-pollination. Okay, benih dia ataupun debunga dia akan pergi kepada tumbuhan-tumbuhan yang natural. Apa kesan transgenic genes ini pergi keluar daripada kebun kita, masuk ke dalam hutan, masuk to other people's place. So there may be impacts, there may be effects that we don't think about when we are doing the research. Okay, so ini adalah untuk dibahaskan, untuk difikirkan, untuk direnung, tidak perlu hafal, sekadar tahu dan sedia maklum lah. Okay, so yeah, so those are the ethical issues, and that is pretty much it for this subtopic. Okay, lebih kurang itu saja untuk subtopic ni dan untuk chapter ini. I'm going to do a quick summary of 8.3. first things first, we learn about the production of cDNA. Mula-mula kita belajar macam mana hasilkan cDNA. And then we learn how that cDNA can be used to produce insulin. We also talk about areas in which we can apply recombinant DNA technology. Kita boleh gunakan recombinant DNA technology dalam bidang penternakan dan pertanian, which is agriculture. Kita boleh gunakan untuk memanfaatkan alam sekitar. We can use it to benefit the environment. And we can use it for research and medicine. Research and medicine is example, insulin. Okay. Not to forget, there are ethical issues surrounding recombinant DNA technologies. And I hope that you would be willing to learn more walaupun tidak perlu hafal, tidak banyak yang kamu perlu ingat. Sekarang saya harap kamu ada jiwa untuk belajar lebih banyak. Okay, like, go and learn something. There might be something new about recombinant DNA technology that is outside your syllabus. I encourage you to go and learn about it if you have the time. Okay, banyak sudah Miss merepek. Thank you for joining me for this lesson. Now, what should you do? You should make your own notes or mind map. Dalam kamu punya nota ataupun mind map yang kamu buat, sila tekankan kepada penghasilan insulin menggunakan cDNA. So, in your notes or in your mind map this time, focus more on the production of insulin and the synthesis of cDNA. After you're done making your notes, please go ahead and complete all the Chapter 8 tutorial questions, and we will discuss them when we have the time. That is pretty much it for me and this chapter. Thank you very much for your time and attention, and I will see you again soon. Happy learning, and bye!